Hello everyone, my name is Ankur and welcome to RJPT Simlab simulation. In this video, we will study about the anticonvulsant property of diazepam against PTZ-induced convulsions in mice. At the end of this video, we will be able to tell about the anticonvulsant effects of diazepam drug on the pentylene-tetrazole-induced clonic convulsions in mice. Now, let us start with the video. At first, in the introduction part, let us know what are convulsions. Convulsions are the uncontrolled rapid muscle contractions and spasms that causes the involuntary shaking of the body. These are the rapid muscle movements and contraction of these muscles and spasms that causes the involuntary shaking. Involuntary shakings are those movements which are not in our control. And these controls are naturally occurred by the body. So these are the involuntary shaking of the body. And what are then anticonvulsants? Anticonvulsants are the medications that we use to prevent or treat these seizures, often by controlling abnormal electrical activity in the brain. So these are the drugs or medications that we use to treat or control these effects, these involuntary actions of the convulsions. And what are clonic convulsions? Clonic convulsions are type of seizures. These are only a type of seizures which are characterized by, we can say, these are observed by their actions such as rhythmic or jerking movements of the arms and legs of the animal or human. So now we know what are convulsions, what are clonic convulsions, and what are anticonvulsant medications. Now let us see the equipments required for this experiment. At first, we need our animal mice, which should be of 20 to 25 grams in the body weight. This is an average body weight of a mice. Then second, the drugs. We need pentylenetetrazole, PTZ. In short form, we call it PTZ, which should be of dose 80 milligram per kilogram. We have to introduce it in mice, and for that we have to prepare a stock solution containing 8 milligrams per milliliter. For 1 milligram, for that we have to prepare 1 milligram per 100 grams solution of according to the body weight of the mice. The second drug we require is diazepam. Diazepam we require a dose of 4 milligram per kilogram, and with that we have to prepare a suspension of with 1% gum acacia at a concentration of 0.4 milligram per milliliter with a dose of 1 milliliter per 100 grams of the body weight. So this is the dose required for the diazepam. Let us know what is the pentylenetetrazole drug. This is a chemical compound that acts as a central nervous system stimulant and convulsant. So it is a stimulant drug which acts on the central nervous system. And this drug is also used to induce neurological disorders and replicate the characteristics of human epileptic seizures in animal models. So this drug induces convulsions. This drug creates convulsions in the humans or animals. And with that, we study about the epileptic seizures occurrence in the animal models. Okay. So now what is diazepam? Diazepam is a benzodiazepine sleeping pill. Diazepam is an anxiolytic, or we can say it, anti-anxiety drug, which is used for its effects such as sedation, muscle relaxant, and anticonvulsant properties. So, with the diazepam drug, we will treat the convulsions that are created by the pentylenetetrazole drug. Okay. Now let's move on to the principal portion. The principle says pentylenetetrazole (PTZ) is a central nervous system stimulant, as we know, known to induce convulsions in rats and mice. So this drug will stimulate and induce the convulsions in the rats and mice. So this experimental model is commonly used to stimulate convulsive episodes for evaluating the efficacy of anticonvulsant drug. So we are performing this experiment for the study of the anticonvulsant effect of the anticonvulsant drugs, and we will evaluate by stimulating the convulsive episodes by introducing the PTZ drug in the rats and mice. So we'll study about the anticonvulsant drugs effect. So the PTZ acts by modulating GABAergic pathways, producing clonic convulsions that mimic seizures activity in humans. So This PTZ, the drug, acts by modulating the GABAergic pathways. What is GABAergic pathway? The GABAergic or GABA pathway is a metabolic process but for synthesizing. It synthesizes and stores and breaks down the inhibitory neurotransmitter GABA. Okay, so as we know, GABA is a neurotransmitter. The full form of GABA is GABA aminobutyric acid. Gamma aminobutyric acid, GABA, and it is a antagonist. It is a neurotransmitter and acts as an antagonist. GABA, the GABAergic pathway starts with the precursor of glutamate, and then it is decarboxylated by the glutamate decarboxylase enzyme to form the GABA. GABA is the gamma aminobutyric acid. Okay, so this produces the GABA pathways produces the clonic convulsions that mimics the seizure activity in humans. Okay. The anticonvulsant drugs diazepam works through GABA receptor modulation, and this is used here to assess its effect in preventing the PTZ-induced seizures. So the anticonvulsant drug, our diazepam, works through this GABA receptors modulation. This this drug modulates the GABA receptors, and here we use this drug to assess its effect in preventing the seizures induced by the pentylenetetrazol PTZ drug. Okay. So this is the observation table that we will draw when we will perform our experiment. We will draw it for two groups, the control group and test group. We will perform this experiment by dividing the animals into two groups, control group and the test group. Okay, now let us see the procedure part. The procedure says that the first step is grouping and preparation, in which we will weigh and number the mices, and then we will divide the animals into two groups, each consisting of five mices. We will divide into two groups and each will consist of five mices. The group one will be called as control group, which will receive the PTZ drug only. And the second group, which will be known as test with diazepam, receives the diazepam followed by the PTZ, which means at first we will give the diazepam drug and then after that we will inject the PTZ drug. Okay. Then second is the drug administration. In the control group, we will inject the pentylenetetrazole at 80 milligram per kilogram. We will inject our drug, and to the test group, we will inject diazepam at 4 milligram per kilogram, then administer the PTZ at 80 milligram per kilogram after 30 minutes. So after inducing diazepam, after 30 minutes, we will administer the PTZ drug. Okay, the third step is the observation. We have to record the following parameters. The first, the onset of convulsions. You have to record the time from PTZ administration to the onset of convulsions, including jerky movements and clonic seizures. We have to record after the PTZ administration into the of the control group. What we have to observe the onset of convulsions, which means the starting of the convulsions, including the jerky movements. In convulsions, the mice or rats will produce jerky movements and clonic seizures. Okay. They will start throwing their arms and start shaking. Then second, we have to measure the duration of convulsion, which means you have to record the duration of clonic and extensor convulsions. And then third, the recovery or death part. You have to note whether the animal recovers from the convulsions or succumbs to the convulsions. Succumbs means succumbs to death, or that causes the death of the animal from the convulsions. Okay. Thus, the next parameter is data collection. We have to record each parameter, onset, duration, recovery, or death for all the animals in both groups. Okay, so now let us perform this experiment. And we will perform this experiment into our RJPT Simlab's simulatory laboratory. As you can see here, this is the UI interface of our simulatory laboratory. On the left side of the toolbar, we can see the cage and the weighing machine. When we will click it, it will be placed on our table, laboratory's table. Okay, on the right side of the toolbar, we can see the syringe, which is pre-filled with our drug solutions. On the bottom side, we can see the instruction part, which will provide us the procedure part, the instruction through which we will perform an experiment. On the right side of that, we can see the next escape button to move forward, and on the top side, we can see the observation table button. This is the drop down button. When we will click it, we will see the observation table over here. This is the observation table of the control group. As we can see here, it shows the serial number, which means we have six animals. We are using six animals. We can use either five or six animals. So we are using six animals in this experiment. We will record the body weight and then the treatment to the control group. We will provide the PTZ drug and then the onset, the timer, and then the nature of the severity, which means the reaction of the animal, and then what happens, either death or recovery of the animal in the according to the timer. When we will click the test group button, we will see the observation table of the test group. Here also is the serial number of six animals, their body weights, and then in the treatment we are providing them diazepam 30 minutes prior, 30 minutes before to the PTZ drug. Okay. So now it also contains the onset timer, nature and severity, and death and recovery. When we will click to the control group, we will see the control group observation table. On this button, procedural flow button, we can see the step-by-step procedure of this experiment from the drag mice case to the to the completion of this experiment. And we can click these buttons to move to jump to any step of the experiment. Okay, we will move it upward and we will start with the experiment. So the instruction says place the cage from the left sidebar. So when we will click it to the cage button, the cage will be placed on the table of our lab. And as we can see, we have two cages. One is of the drug group and second is the control group. Then the instruction says place the weighing machine from the left sidebar. We can even hide these sidebars by clicking this hide and show button on both the sides. Okay, so now we'll place the weighing machine. As we can see, the weighing machine is also placed and now we have to weigh and mark each mice from the control group. So we will start weighing the control group mice. One by one, we will weigh it. First, we will mark it with the marker and then we will place it in the weighing machine. As we can see here, we will weigh and then again we will place it into the control group's cage. Again, we will pick our second mice and we will place them and we will mark them as second number. The two line indicates the second number and again we will place it in the weighing machine and then again in the cage. Similarly, we will perform this for each and every mouse. As we can see here, we will mark them. Three line says third number mouse. We will weigh it. As you can see, it was of 20 grams. And as we know, the average weight of mice can be of between 20 to 30 grams. We will mark this one as the fourth. And when we will see the observation table, the body weights are being entered over here into the table. As we can see here, when we will weigh it, automatically the body weight will be written over the observation table. This is our fifth animal, our fifth mice. We will mark it with the five lines. And when we will place it into the weighing machine, the weight will be recorded over here. And this will be our last mice. We will mark it with six lines. You have to carefully mark it so that no lines intersect. So otherwise it will create a misconception. Again, we will place it into the control group. Now, after performing this, we will move on to our next step. We will weigh and mark each mice from the drug group. Now, again, similarly, we'll start with the drug group. We will weigh, mark and weigh from the drug groups. By clicking the skip button, we will move directly to the next procedure. As we can see, this is our control group and this is our test group observation table. So just like from the control group, the test group is, we are performing the same for that. The body weight is being entered over here automatically when we when we weigh the weight of the mice. When we click the skip button, it will directly move to the next step. As you can see here, when we click the skip button, all the body weights are being measured, and we have moved to the next step. The next step says we have to place the syringe from the right side bar. When we will click to the right side bar, the syringes will be placed over here. As we can see here, we have two sections, pre-filled diazepam and pre-filled PTZ. So at first, we have to apply the Pentylenetetrazol PTZ to the control group and take the reading. So we will click the apply syringe button and one by one we will inject our PTZ drug. So these are the six syringes for each and every mouse. Slowly we will inject it and we will place it over this board. This tray contains numbering for each and every mouse. For the second mouse, we will inject it through the intraperitoneal route and then we will place it to the second number. As we can see here, when we are placing the mices on the numbers, the timer has been started for each mice for the recording of the onset of action of the PTZ. As we know, the PTZ induces the convulsions in the mices. As we can see, the timer for each mice has been started when they're placed in their position in the tray. As we can see, the time is being moved, and one by one we will place each mice in their positions, and we will not reuse these syringes because it can cause a chance of infections to these mice and it will hinder the experiment. So we will use different syringes for each mice. The time is running, and we will inject our pentylenetetrazole drug to the last mice of the control group, and we will place it on the sixth number in the tray. And now we will observe the reaction of these mice. Slowly, these mice will show started convulsions. As we can see here, the first one has started to move, and its body is shaking. It's moving forward. The second mice has also shown its onset of action. As we can observe, they have lied down to the tray. They are shaking their legs. The fourth one has started to move. It's moving forward and its body is shaking. The fifth one has also started. The time of this onset of the convulsions are being recorded. For the first, it's 55 seconds. The second, it's 72. The third one has shown convulsions in 62 seconds. The fourth one has shown in 75. The fifth one in 60 seconds. And the sixth one in 67 seconds. So these are the timers for the convulsions, for the onset of convulsions. When we will drop down the table of the test group, basically of the control group, we can see the reactions. So when we see that the first one has shown its conversions in 55 seconds, so the nature was jerky movement and strop tails. The mice has shown jerky movement, which means shaking of the body, and strop tails. Strop tail is a condition in which the mice carries its tail in an erect or nearly, basically we can say, vertical position. And as we can, in simpler terms, we can also say that it is an abnormal tail movement. When we have seen that the mice was moving its tail in an abnormal way, and the body was shaking of the mice. The second mice has shown clonic convulsions. As we know, clonic convulsions are the movement and shaking of the hands and the body. The third one has also shown clonic convulsions. The fourth one has shown jerky movement and strop tail. The fifth one has also shown the same jerky movement and strop tails. And the sixth one has also shown clonic convulsions. So the death and recovery part says that the death and recovery of the first one is 3, 5, 4, 3, 5, 3. And now we will move on to our next step. We will save these observations and move on to our next step. We will slowly place these mice back into their control group's cage. And then we will move on to our next step. We have to perform this experiment very carefully and this step very carefully so that no mice gets hurt. Now we are placing our last mice in the cage. And now we'll move on to our next step. In the next step, we have to apply diazepam to the drug group. While clicking the apply syringe button, we will apply the diazepam drug to the drug group. Similarly, with the pre-filled syringes filled with diazepam, we will administer these diazepam drug through the intraperitoneal route. We will apply our drug slowly, one by one, to each mice through the intraperitoneal route, and then again place it in the drug group's cage. We will perform that for one by one. So we will skip this step and move on forward. And now, after doing in administering the drugs to all the six mice, we will wait for 30 minutes for the diazepam drug to take its effect. The diazepam takes 30 minutes for it to show its effect. We will skip this timer and then move on to the next step. Now we will apply the PTZ Pentylenetetrazol drug to the drug group and take the reading. By clicking this apply syringe button, we will administer each mice one by one from the serial number and administer the pre-filled syringes filled with the PTZ drug through the intraperitoneal route. And then we will place them on the table, similarly as we have done for the control group. The timer has also started for the first mice, as we can see here. Similarly, we will do it with the second mice also. We will inject our PTZ drug through the intraperitoneal route and then place it on the second position on the tray. The timer has also started for the second mice. As we can see here, we are applying the PTZ drug one by one carefully to all the mice and placing them on their position tray in the tray. And the timer has started. And then again here we will observe the onset of time. As we can see here, the table of the test group shows the onset of time in seconds and the nature and severity and the death and recovery according to the minute. We will administer our drug to the fifth mice. The timer has started for all the four mice that we have placed on the tray. And now we have placed the fifth mice. And now we will administer our drug, PTZ drug, through the pre-filled syringe to our last mice. And after that, we will observe the effect of the PTZ and the effect of diazepam on the mice. The PTZ will induce the convulsion activity and the diazepam will act against it in an antagonist way and reduce the effects of PTZ or convulsions. As we can see here, the sixth and the fifth mice has shown its action on the 120 seconds, both has shown on the same time. And similarly, we will wait for other mice too. So now when we have done this, we will save our observation. And now again, we will place them into the drug groups cage one by one. As we can see here on the observation table, the first mice has shown no convulsions, the second mice too, the third and the fourth. These four mice has shown no convulsions, and the death or recovery time has not been shown because no convulsions were induced in these four mice. But in the fifth and sixth mice, convulsions have been shown. Oh, but slight convulsions, we can say clonic convulsions are the small convulsion activity, and it had been shown in the second 2 minutes after the 2 minutes of the after administering the drug. So by clicking this, we can see and compare both this control and the test groups at result. And now we'll see the result of this experiment. The results is in the result, we can see the observation table of both the control and the test group. The control group has shown has the control group has shown that each mice has shown jerky movements and stop tails and tonic convulsions after inducing convulsions in them with the PTZ drug, and has shown the death or recovery in the third minute, fifth, fourth, third, fifth, and third minute for each mice. But in the test group, after inducing diazepam with the PTZ drug, four mice has shown no convulsions, no sign of any convulsions, but the fifth and sixth mice has shown slight convulsions or clonic convulsions, but even after 2 minutes. So the diazepam, so with this experiment, we see that the diazepam reduces the convulsions activity. It acts as an anticonvulsant and reduces the convulsions effect. It slows the abnormal and excessive nerve activity and reduces the convulsions. The convulsion causes the anxiety in the animals and panic attacks, but the diazepam acts against it and shortens with its treatment. So for the result, we can also calculate its mean calculation. We can calculate its mean and standard error of the mean. for each parameter in the both groups. And we can also compare the latency to onset, duration of conversion, and recovery rate between the control and the diazepam treated groups, as we have discussed here. And a significant increase in the latency, reduction in duration, and or increased survival rate in the diazepam group suggests anticonvulsant efficacy, which means a significant increase in latency in the death or recovery time, reduction in the duration, as we can see here, and an increased survival rate of the diazepam group suggests anticonvulsant efficacy, which means diazepam has shown anticonvulsant effects. So, I, so here we can understand the effects of diazepam, anticonvulsant effect of diazepam on the mice. So here we have come to the end of this experiment. I hope you have learned a lot about the anticonvulsant effect of diazepam and the effect of PTZ inducing convulsions in the mice. So guys, now we have completed our experiment. Thank you guys for watching this video. And for more such learning content, subscribe to our YouTube channel RJPT Informatics. And you can also follow us on our Instagram profile, on our LinkedIn profile, as well as in Facebook. We update and upload our such learning content over these platforms. Thank you.